Uh, um, Hi everyone, thank you for joining me. So today I'm going to talk about my work on integrative omics for the discovery of biosynthetic pathways using MEANtools. Um, to start with and to give you a little bit of background, plants produce a wide array of natural products, and sometimes natural products are also called as specialized metabolites or plants chemicals. So I will stick to natural products. And these natural products, and we as a human actually heavily rely on those natural products for flavors, fragrances, drugs, agrochemical products, etc. But these compounds, however, they represent the tip of the iceberg as far as the total structural diversity displayed by the plant kingdom. And these plant specialized metabolites, which are responsible for the biosynthesis of those natural products, sometimes are encoded as gene clusters. And they are also known as biosynthetic gene clusters, so like bacterial word. So this term has been adapted from prokaryotes, but in plants, biosynthetic gene clusters are rather an exception rather than as a rule. So sometimes you see these clustering happening, and sometimes they're not. But the discovery of Dimboa was the first biosynthetic pathway which was identified based on gene clustering. And since then, there are like 30, 40 pathways that has been discovered based on this property of gene clustering. But gene clustering gives you just information about the genes which are clustering together and all on the genomic loci. But you don't have, if you don't include any further information like the expression information or metabolomic information, it's really difficult to identify the pathway behind the biosynthesis of that particular natural product. But there is difficulty in accessing those natural products, like in terms of finding genes and metabolites, because of several reasons. First, they are lineage specific. So biosynthetic genes, sometimes they're present in very specific orders, very specific clades, and it's really difficult to identify the pathway or other genes using comparative studies. And the gene expression and the abundance of those metabolites are really dependent on certain growth stages, and they are also present sometimes in particular tissues, or those pathways are triggered in response to certain environmental triggers, which are difficult to reproduce. And if we talk about the concentration of those metabolites, the concentration of these secondary or specialized metabolites are really low if you compare it with primary metabolites that are present in plants. So we have recently published a review paper where we have timeline the discovery of these biosynthetic pathways. So since the discovery of first Dimboa pathway, and then we have highlighted that those discoveries using the omics they have used to discover or to identify those pathways. And you can see a clear trend. So since the discovery in 1997 that researchers really adopted single omics technologies, and then in the recent years, last 5 years, we see really adoption of multi-omics approaches to identify those biosynthetic pathways. So in the NVO Root Consortium, we are mainly interested in how plants recruit beneficial microbes, and that's why we are really interested in the plant-specific pathway or the mechanisms behind the recruitment of those specific pathways. And that's why we are integrating this multi-omics or integrative omics approach, where we are kind of using all three omics strategy, and it has proven so far really useful for the elucidation of novel biosynthetic pathways. So talking about the methods which are available, I think one way to compare if you have transcriptomics and metabolomics data is like you see the expression of genes. If you have transcriptome from roots and leaves, you can immediately see the expression and abundance of metabolites and compare it. The second way is to match the chemical transformation. So you create a co-expression network from your transcriptomic data, and then molecular family network based on chemical transformation, and then compare it. And the third way is separately with transcriptomics data, you perform KEGG enrichment and find out the classes of metabolites present in your transcriptomic data. And the same way you classify the metabolites using independent tools like classifier, and then compare it. So to this direction, actually, we have developed a tool called MEANtools, and this is based on the chemical transformation, rather. So what we do here is we in the first step, we correlate transcriptomics and metabolomics data, and also in the second step, we integrate all the publicly available knowledge present as reactions in the databases, and also different structures present in the structural databases, and then identify biosynthetic pathways or chemical reactions. So the input is, so you start with metabolomics, you perform your sample extraction, do this LCMS or LCMSMS, generate your raw spectral data, and do some pre-processing, and you get to this stage where you have a feature table where you have rows metabolic features, and then as columns you have the abundance of those metabolic features as different samples. So you start with this, and same with transcriptomics. You start with the raw sequencing data, you generate your quantitation matrix, where you have genes as rows and then their expression in different samples and in different columns. So these are the two tables you need to start with. So first step is correlation. So we have provided different methods for correlation, like you can use either a simple correlation method, Pearson correlation, Spearman, or Pearson log, but we recommend to use a mutual rank correlation because it was very effective in one of the research paper published by Jennifer Weisgeber recently, where she actually generated co-expression network based on mutual rank. And yeah, creating mutual rank is actually, the background is correlation. So you do correlation, and then instead of setting a threshold, you just rank the correlation of a gene versus another gene if you're just using transcriptomic data, and it's just then a simple geometric mean of the correlation between a gene and then in the reverse order. And the another thing is, so with mutual rank, the number goes really high. So it depends on the number of features or the number of genes you have. So it could be anything between 0 and the number of features -1. So if you have like 1,000 features, then it could the mutual rank can go up to 999. So you really want to use that mutual rank statistic to generate a network, and that's why she used the exponential decay function to convert that big mutual rank number into a number between 0 and 1. So you convert that number and use that as edge score, and then generate a network, and then you can use any clustering tool to generate clusters, and then edges would be your edge score, and the nodes would be your gene. If you are using just transcriptomics, it would be gene versus gene, and if you are using transcriptomics and metabolomics, then it would be the nodes would be gene connected with a metabolite. And that's what we actually wanted to see, because it worked really well with the simple transcriptomic data. With the co-expression, she in the paper, Weisgeber paper, she really identified all the biosynthetic pathways where genes present in the pathways, they all cluster together, and not only biosynthetic genes, but the transporters and the regulators, they all form a functional module. It really worked well with co-expression networks, but we really wanted to see if we mix this with metabolomic data, whether we would be able to identify gene metabolite a functional network using this approach or not. So I'll talk more about that when I show you the results. But then the second step of the MEANtool is the parsing of databases. So MEANtools uses two databases mainly, RetroRules, which is a reaction database, and it has around 43,000 reactions. It gives you some information about different reactions, and also some meta information like the EC number, and also most importantly the UniProt ID. So the UniProt ID gives you the information that which enzyme is actually catalyzing the reaction. And with this UniProt ID, we have actually cross-linked this UniProt ID with other databases like KEGG Orthology and RIA databases. So including this 43,000 reactions, we have extended the search space to around 60,000 reactions by cross-referencing these IDs to other databases. The second database, what MEANtool parses, is NPDB. So this was the work of a MSc student in Wageningen University. He compiled all the publicly available structural databases together and built one SQLite file where you have all the information. And MEANtools uses this. And this one is publicly available, so you can download it if you want. So I've provided the link in case you just want to test it. So these are the two databases mainly which MEANtools parses to get to a stage. So talking about some functionalities, so how it calculates novel reaction, it's like everything depends on mass transition. So let's just say you have a reaction where you have a substrate and a product, and a substrate has certain mass and a product has certain mass, and mass transition is just the difference between these two masses. So what MEANtools does is like, So you have a gene metabolite correlation, which you calculate using mutual rank, and then MEANtools uses NPDB to assign all the metabolic feature a substrate, because then you can really putatively annotate your metabolic feature to a certain compound. And then using RDKit, it predicts the product of those substrate. And once you have this substrate product information, it calculates mass transition from your metabolomic data, and it already had mass transition calculated from the known reaction of the databases, and then it compares these two mass transition between your metabolomic data and the reactions which are present in the publicly available database. And while doing this, it also generates a mass degeneracy table, and this is quite informative, I would say, because on x-axis you have mass transition both in both directions, plus and negative, and on y-axis you have number of reactions. And by seeing this, you can immediately spot how many reactions can be attributed to a particular mass transition. So, so this is the main fundamental behind the working of MEANtools. So MEANtools calculate mass transition, compares it with the database, the publicly available databases reaction, and then predict novel reaction. So the use of transcriptomics is just to prioritize your metabolic features, because normally when we create a feature table, you get to a stage where you have 1,000, 2,000, probably sometimes 20,000 features, and you have no idea what to do with that. So using transcriptomics, you find good correlation between genes and those metabolites, and then you can use it further to predict reactions. So talking about a bit of summary workflow, so it's quite modular in nature. It has been written in Python. So the green module is all the correlation module, where you have scripts for the correlation, and you have multiple options available there. The blue one is for the database parsing. It actually pre-processes. It doesn't use as it is. It pre-processes the whole database and makes it available so that it can be further used in the later stages. And then it has a prediction module where it compiles all that information, the reaction and the correlation, and then it predicts novel reactions. So what we did was, so there was a study published in 2020 by Yon et al. And they characterized a novel modified fatty acid pathway called falcarindiol, and it is antimicrobial, antifungal metabolite. And what they have done is they have used the paired omics data set. So we always stress on using paired omics if you are really interested in identifying biosynthetic pathways. So that means both transcriptomics and metabolomics are coming from the same source. And then they have identified the gene cluster responsible for this falcarindiol metabolite, and they have also characterized all the 8 structures and 7 steps of this falcarindiol pathway, which leads to the biosynthesis of falcarindiol. And what we wanted to see is, like, if we use MEANtools using this publicly available data set, whether we would be able to reproduce the same pathway without going through their steps. So we used the mutual rank correlation in the first step, and we identified a functional module which actually harbored all the 4 genes present in the gene cluster, but it also has the gene which was not present in the gene cluster, but it was present in the pathway, that the last step, the P450. So this was quite interesting, because none of the method gave us this kind of results, and mutual rank really worked in this way. And we use mutual rank in this network or clustering method, not just transcriptomic data, but we also included metabolomic data. So all the green nodes you see outside, and this one, they are the genes, and all the colored nodes, they're the metabolites. So, and then we try to iteratively annotate the metabolite, and we found most out of 3, like 3 out of 6 metabolites belong to lipid classes, and that's what we were expecting, because it's a lipid-based pathway. And then we went ahead and carried out the whole reaction passing step, and in the end we identified proper, so this is the output of MEANtools. It gives you a network-like structure, and also a final representation of all reaction in a reaction format, where you can see that individual structures, they are annotated with the metabolic features which represent those structures, and also the gene or enzymes which are responsible for the catalysis of those intermediate steps. So this is the final output. So you don't get just this reaction, you get all other reactions as well, because if you are treating a tomato plant with the pathogens or microbial pathogens, it is not only falcarindiol, but other pathways get triggers as well. So we also identified other pathway, but because we were focusing only on the falcarindiol pathway, we just reconstructed the whole pathway using MEANtools. So that's what we did. But this, I just want to mention that this data set was MS1, not MS level. We could only do the putative annotations. And what we are doing at this moment is we are trying to rigorously test MEANtools with deep data set like LCM, SMS data, and also deep transcriptome data, so that we can, once we find those functional modules, we can immediately see what kind of structures those are in a very confident way. So that's what we are doing. So in the end, I would really like to acknowledge all the contributions. So Manik, Justin, and Elizabeth for the initial supervision and algorithm design. Hernando and Olga for the initial development, and thanks to our collaborator for sharing the paired omics data, because that's the must thing what we need to test MEANtools, and the NVO Group Consortium for getting the funding to carry out this research. And thank you all again for the attention, and I'm happy to answer any questions you have. Thank you.
All right, any questions?
So this was the first time I heard about the NPDB, and I know there are other resources nowadays like Lotus, for example. Could you just comment on, is the NPDB best or?
No, it's not best. It's very redundant, because it just the student compiled all the databases and put it in one place, and it is a SQLite database, and it's not even published. It's just a link in a Dropbox, so it's available there. But we have made it like quite flexible. So if you have your own customized database, you can use that. And we have also made arrangements for Lotus as well. So if you want to use Lotus, you can use Lotus as well. So it's not like fixed that you have to use NPDB.
Thank you.
How many, now you showed us one example, how many of those data sets with larger pathways are available to kind of develop MEANtools and validate its predictions? Because I think like the data is maybe still a bottleneck.
Yes. Yeah, so not many. So recently there was one study came from Sarah O'Connell lab from Max Planck. They published a paired omics data. It was single cell as well. So we are into collaboration with her, and she shared the data with us. And then this data set, and not many actually, you're right. So we are really stressing a lot, everyone who has this kind of data, so that we can test. But in the NVO Group Consortium, we are also doing experiments. So we will have our own data very soon, because we, as I mentioned in the talk, that we are interested in investigating how plants recruit beneficial microbes. So we are generating paired omics data from leaves and roots upon pathogen infection. So that will be available soon. So yeah, and the worst part is it's really difficult to generate a simulated data. Of course, you can do transcriptomics, but I'm not sure whether you can do metabolomics simulation. So yeah, it's really a bottleneck at this moment. Yeah, true.
All right.